Okay, we'll get started in a few minutes. So everyone takes their seats. Well, welcome back to the afternoon session of the public meeting on HIV patient-focused drug development and HIV cure research. We had a great morning discussion, and we look forward to a great afternoon discussion. My name, for those of you who are just joining us, my name is Sarah Eggers, and I'm from CEDAR's Office of Strategic Programs, and I'll be facilitating much of the discussion today. Before we get to the discussion, we'll have a few welcome remarks, and then we'll set the context for our discussion, and some of my FDA colleagues will present some background information. And then we'll get into discussion. I'll go over a bit about the discussion format after that, before we do. And with that, I would like to turn it over to Dr. Janet Woodcock, who is the Center Director, for a few comments. Thank you, Dr. Woodcock.
Thank you, Sarah, and good afternoon, everyone. It's great to see the folks who've turned out for this meeting, and I hear there are a lot of people on the web. That's fantastic. I especially want to thank the HIV patients and patient advocates for sharing your experiences and your perspectives on this disease and on the therapies. Today's meeting is part of an initiative that we're running that's called Patient-Focused Drug Development. We are quite excited about this. Teresa Mullen gave an overview this morning, but I'd like to reiterate some of her points, especially since some of the folks were not actually able to be here this morning. As Teresa mentioned, patient-focused drug development was an important aspect of the package for the 5th authorization of the Prescription Drug User Fee Act, or PDUFA 5. And it's an enhancement of our current mechanism for getting patient input on important issues regarding their diseases, which have tended to generally focus on specific drug products, and we want to focus more broadly on the disease entity. Patient-focused drug development, therefore, is a broader, more systematic method of obtaining patients' perspectives on the severity of a condition and its impact on daily life, as well as their perspective on the range of available treatment options and the impacts for good and for ill of those various treatment options. And we recognize that even within a disease, and some diseases this is more true than others, there's a range of severity that people are experiencing, often from being relatively well and functioning to suffering severe effects from the disease or from the treatment, in fact. And we want to hear about that. We want to hear about the burden of disease and the burden of therapy. It's valuable for our drug review because it helps provide the clinical context in which we evaluate new treatments, all right? Because we don't do that in a vacuum. We say they're safe and effective, but really mean the benefits of a new treatment outweigh the risks, and therefore you have to understand the disease, its risks, and the effect of the current treatments, and really do that, I think, partly from the patient's point of view as well as from the medical point of view. Patient input can help drug development efforts more broadly also by highlighting where new types of outcome measures are needed to help make sure that new therapies treat the symptoms that most matter to patients, or they avoid side effects that are most important to patients. And often we don't put all of these outcome measures in the trials, so we may not know about certain important impacts on patients. For example, for many diseases, we have never learned anything about impact on sexual function of some of the treatments for various diseases. And some therapies have very profound impact on sexual function. And if you don't ask about that and record it, then you never learn. And people simply have to experience that. So, but more broadly, we'd like to have outcome measures that we devise for benefit as well that are really relevant to patients and patients feel are the right outcome measures for that disease and the burden of disease that they experience. So this is an ambitious undertaking, I think, and part of our commitment under PDUFA that we made to Congress. We will convene at least 20 public meetings over the next 5 years, with each being focused on a different and specific disease area. And today is our second meeting within this initiative. Our first meeting was on April 25th, and it focused on chronic fatigue syndrome and myalgic encephalomyelitis. It was, from our point of view, very valuable, and that's a disease that really doesn't have any approved treatments at all, and who has very profound effect on the most severely afflicted people. We received a lot of positive feedback from participants in the patient community who attended that meeting. They felt it was very helpful. And in that disease, we're really seeking to attract drug development and understand what outcome measures would be most appropriate in that disorder. This morning's discussion here focused on the most significant symptoms associated with HIV infection or its treatment and what impact it has on daily life. We also heard your perspective on the currently available therapies to treat HIV. This afternoon's discussion is a bit different, and this, I think, each meeting we have will have different flavor. It will focus on some important issues with respect to cure research studies. In the past, HIV cure did not seem possible, but today researchers are looking at new ways to either clear HIV from the body or control the virus without the need for antiretroviral therapies. One of my colleagues is going to provide more background on what we mean by cure research. But understanding the patient's perspective on the potential benefits and risks of participating in HIV cure research studies will help FDA evaluate drug sponsors' protocols for clinical trials and their informed consent procedures. So this is very exciting, I think, for HIV, and shows that in this disorder we're at a more advanced state of treatment, say, than we are with, say, chronic fatigue syndrome, where we really don't even understand the etiology and we have no available treatments. So thank you again for your valuable time and input on HIV symptoms, currently available therapies, and I wish you luck this afternoon. I hope we have a very good discussion on both the ethics and the feasibility and desirability of HIV cure research. Thank you very much.
Thank you, Janet, and good afternoon, everyone. My name is Richard Klein. I'm the Director of the Patient Liaison Program at FDA in the Office of Health and Constituent Affairs, formerly known as Office of Special Health Issues. And I just wanted to do a brief overview of the main thoughts, at least that I heard this morning, and try to summarize what we heard. To me, one of the main issues that came up and kept coming up was comorbidities, people dealing not only with HIV, but with HIV's concomitant problems and other diseases. So it had a lot of ramifications that kept coming up through the morning, managing hepatitis, managing hypertension, managing your drugs, and drug interactions. And drug interactions was, I think, the second most common thought that kept arising for me this morning. People dealing with multiple drugs for multiple conditions and how to work those drugs together and deal with the drug-drug interactions and finding the right drug combinations. I think e-prescribing was raised as one potential way to address those kind of problems. Could people have electronic records that would help guide and prevent the use of drugs that shouldn't be used together, that are contraindicated, or that had drug-drug interactions? Adherence and ease of dosing was another issue that came up this morning. And that led to another question about challenges of changing meds. Were people comfortable with simply changing meds when you had a workable, successful therapy? Would people be able to switch to something else for ease of use and better adherence? And people were a little reluctant to do that. If they're successful, it's kind of, if it's not broke, don't fix it. Unanticipated consequences, I think people were talking about hesitancy of changing drug regimens also because of the fear of switching regimens and then using up something that would have been a future option for you to use. And so people didn't really feel comfortable with the idea of burning through different classes of drugs. So changing regimens is a difficult decision that people were dealing with. And that question about using up scarce options for future. Whether HIV is a manageable condition was a question that came up, and that differed from person to person depending on whether or not you thought that the problems that you were having related to your therapy and to your HIV were part of the question. So if you could manage HIV but still had all these adverse events from the drugs, was that a manageable disease? I think we were pretty well reminded that stigma is still a problem, that people are worried about diagnosis, talking about their HIV diagnosis, talking about even being on therapy, and that stigma remains a problem. And finally, reminded about the fact of transmissibility of HIV and whether or not being a chronic but manageable disease could affect whether or not people were going to be, whether or not prevention could be impacted by the ease or the alleged ease of treating the disease. And the question about whether or not that could possibly undermine prevention efforts. And I think those were the top issues that had occurred to me this morning. I don't know if other people had other points, but if not, then we want to shift gears for the afternoon, talk about the future, about cure research and where that is, where it's going. And for a background on cure research, I want to introduce the chief of the General Medicine Branch, Division of Clinical Education in Pharmacology and Toxicology, the Center for Biologics. And his name is Ilan Irony. And with that, I turn it over.
Hi, good afternoon. Again, my name is Ilan Irony. Thank you very much for the introduction. I'm from CBER, the Center for Biologics Evaluation and Research. And so this is an outline of my brief introduction to the subject of HIV cure research, and this is the background. So I'm basically going to describe the FDA organization and our particular place in the FDA organization, our center, in our office. Talk about the strategies for research that aim towards a cure of HIV infection. We'll talk a little bit about gene therapies as they pertain to HIV cure research, which is part of what we do in our office, in our center. And then wrap up with combination strategies and the ideas and the controversies in combination strategies in also HIV cure research. So first, a word about the general FDA organization. We're all under the Office of the Commissioner of the FDA, and the list here contains a list of the centers. Each center is particularly responsible for an area of human health or animal health. But I wanted to emphasize the last three centers here that pertain also to HIV, human health related to HIV. So the Center for Devices and Radiologic Health, Center for Drug Evaluation and Research, which you heard from representatives this morning and from Dr. Woodcock. And in red here in the last Center for Biologics Evaluation and Research, where Dr. Wheaton and I here in the panel work. Within the CBER organization, there are multiple offices that support the operations of the center, but I also want to emphasize the three offices that pertain particularly to HIV treatment and HIV research. one of them being the Office of Blood Research and Review, which regulates the blood supply of the nation and tests for HIV, in vitro tests for HIV, Office of Vaccines Research and Review, which regulates the development of preventive vaccines for HIV to prevent transmission of HIV. As we heard in the morning, there was some discussion about the unmet need in terms of having a vaccine, successful vaccine for HIV prevention. And finally, in red here, in the last bullet, the Office of Cellular, Tissue, and Gene Therapies, which is our purview of what we regulate and what we approve in products that are primarily for gene therapies and cellular therapies. So why Cure Research? We heard a lot about this morning about HIV infection being transformed in the last 30 years from a manageable chronic condition, from a lethal condition into a manageable chronic condition. And that's debatable how manageable it is. We heard a lot about the comorbidities and the symptoms that remain. So the fact that it's not lethal doesn't mean that there is no unmet need for patients and caregivers and for society in general. But we have to recognize that it's been a big progress over the last 30 years in terms of transforming a lethal disease that quickly progressed to AIDS and opportunistic infections into something that people can live through medications. But still, there are some drawbacks to this. There is a lifelong requirement for antiretroviral therapies with both long and short-term side effects of those therapies, of those drugs. And we heard a lot about them, about effects on comorbidities, effects on other medications that people need to take because of complications of those antiretroviral therapies. So there is a need that FDA perceives and the community perceives of cure research. And even with very low viral loads, when you have very low or even undetectable viral loads, we know that there is some degree of viral replication that persists and some latent viral reservoir that's present. And it's debatable where the complete knowledge of this viral reservoir is, but we think that the gut, the brain, bone marrow are places where HIV hides, and as soon as there is a lack of drug, HIV can flare, can result in reinfection and manifestations. But recently we had some, in the last few years, we had some isolated examples that pointing us towards the possibility of a cure. So we have Timothy Brown, known as the Berlin patient, or the Visconti cohort, or more recently the Mississippi baby, that point to the possibility that some patients can be cured from HIV infection. And as Dr. Cox alluded to early in the morning, our definition, our working definition here of cure is either elimination or control of HIV infection without further medical intervention to maintain health. So what are the research strategies that we have seen in development? One approach is, in the first bullet, is shock and kill. Shock we define as activation of the virus from reservoir, from this where it's latent, where the virus is sleeping, to be awakened, to be manifested, and then followed by a kill. So clear the viruses from the body using both boosters of the immune system plus an antiretroviral regimen that's effective to clear the virus. Another approach would be the induction of resistance to HIV infection. And that, in theory, can be accomplished by transplantation of immune cells, CD4 cells or precursors of CD4 cells, from donors that are resistant to HIV, known to be resistant to HIV, or through gene therapies that modify some cells to make them resistant to HIV and then transfuse them to a patient with HIV. And a third possible approach is some experimental drugs or vaccines, therapeutic vaccines aimed to treat rather than prevent, to stimulate the patient's immune system to recognize and eliminate HIV. So that's the reality is that there are lots of challenges in cure research. One of them is for FDA, we need to look at each development step about the protection of subjects in human research. So safety is paramount for that. And we need to ensure that there is a good balance of risks and benefits. So the risk to the patient versus the benefits to patients and benefits for society in general for that research to be accomplished and to yield some results. In those the balance of risks and benefits of research interventions, they change over the course of each product development. So as a product moves along the development from phase 1, first in human study, all the way to phase 3, there's a change in the notion of how much we accrue in terms of knowledge about the product and what the risk-benefit balance is. And as we learn more about parallel development of other therapies or new approved therapies, for example, how do that impact the risk-benefit for that particular patient in that particular research study? There's a challenge also in defining what is sterilizing cure. That means no detectable virus, because as you know, the assays for what's detectable virus and what's the limit of quantification of that has changed, and whether we consider a fragment of a virus a detectable virus or not. Those are debatable issues in terms of sterilizing cure. Equally, there is some debate about functional cure. So functional cure we define as no evidence of disease and no ability to transmit HIV infection. But it's very hard to validate those measures, so the endpoints, as we refer to in clinical trials, for each type of cure to make this a valid endpoint for research and for clinical trials. So let me talk a little bit about gene therapies and as they pertain again to HIV cure research. So first, there are some challenges with manufacturing. The production is usually relatively slow compared to manufacturing of a small drug, and identification of the gene and subtype, and the general manufacturing issues are more challenging than for a small drug. The use of retroviruses or lentivirus gene carriers, those are the buses that carry the gene of interest that you want to insert in humans. They allow for prolonged gene activity. So it's not like you can turn off the effect by stopping a pill, for example. It's another challenge. Usually the product activity, if present, is from a gene-modified cell that needs to survive and function in the patient. And the goal here is to raise resistance to HIV infection. But for that cell to survive, that gene-modified cell to survive, you need to use some busulfan or chemotherapy or some other drugs that are risky to try to make room for these cells and improve the odds that more and more of those cells survive and function and provide resistance to infection. And then there is also the issue of the long presence or long duration of activity, perhaps a lifetime, for those retrovirus or lentivirus gene vectors that carry the gene of interest to the human being. So there's an inability to withdraw or discontinue the product when adverse events occur. And they have also concerns about late onset risks, such as cancer, malignancy. I'm going to talk also about the combination of strategies. So people in academia and industry recognize that it's likely that if there is a chance of success for curing HIV, it will come from a combination of multiple different approaches and to attack the different mechanisms of HIV persistence. For example, the low-level replication or the reservoirs of HIV. It recognizes that combinations with these different approaches may be needed. But there are some scientific issues that are associated with this approach of combination therapies, particularly when we include investigational therapies in the combination. So if it's two approved products and we're combining them, it's a little less challenging because we know a lot about those products. But when one product is new in humans and investigational, it creates some challenges in looking at the combination therapy. One of them is how much information we need about both the safety and the effectiveness of each of the components alone in the course of this development. The recognition that if a combination therapy is needed, each component alone may not work to cure HIV. So patients may be at risk of this drug, but they are unlikely to benefit from research from each component alone. So that raises some informed consent issues, and Dr. Goldkind is going to be talking more about this. And for topic 2 for the afternoon, those are the basic questions, and we're going to hear them again and discuss them individually during the course of the afternoon. So I'm not going to go over them in detail now. But basically what we want is to know what you perceive as being the benefits of participating in research, what factors you want to consider when you decide to participate in research for HIV cure or not, and what kind of risks you would like to, you would accept or not accept to take into your decision to participate in HIV cure research studies. And or if you are required to stop in your antiretroviral therapy, which makes you stable and relatively healthy and manage your chronic condition, how would that affect your decision to participate? And finally, what else do you want FDA to know about HIV cure research? So this is my contact information. I just want to emphasize that in the last point here is the Learn webinar series from our Office of Cellular, Tissue, and Gene Therapies, which is available in the FDA external website and provides a little bit of our thinking about general product development and issues in pediatric research or some of the challenges we face in clinical trials with our products. And this also some of the websites of public access to the Center for Biologics and Evaluation Research. Thank you very much. With that, Dr. Goldkind.
Good afternoon. So I'm Dr. Sarah Goldkind. I'm the Senior Bioethicist here at the FDA. And I'd like to talk to you today about issues related to informed consent and provide some background for our future discussions, because we would really like to get your input on a number of different issues related to that are impactful on being able to accomplish an informed consent process in a way that's most useful to patients who would be enrolled in clinical trials, which I'll call participants in this presentation. And before I get started on my presentation, just by a show of hands, I'd like to see how many of you have been enrolled in a clinical trial before, a clinical trial of any sort, and have gone through an informed consent process. Well, that will be great. I'm really very, very interested in hearing the input from those of you who have experienced an informed consent process and those of you who are naive to one of those processes. So just by way, I'm going to very briefly summarize the informed consent process for you today. And then I'm going to discuss, out of the 15 or so elements of informed consent that are required under FDA regulations, I'm going to discuss 3 of them in specific. And then I want to talk about a particular issue called therapeutic misconception, which I'll define for you during my talk. And then finally, outline a number of the questions that we would like to have your input on during our discussion session. So first of all, what is an informed consent process? Ideally, what it should be is an ongoing educational process between the investigator or another qualified individual that the investigator appoints and the potential or the current participant in the trial. And a summary of the process and what it should really accomplish is that there will be adequate disclosure of information to allow for an informed decision about participation in the research. There should be adequate comprehension of the information that's presented, an adequate opportunity to consider whether or not to participate. It shouldn't be done in such a hurried manner that you really don't feel like you had ample time to consider the clinical trial and its ramifications, to talk to trusted family or friends, and then finally a voluntary agreement to participate. But the informed consent process doesn't end there. It's more than just signing the informed consent document. It should really be a continued disclosure of information as the clinical trial progresses. There may be new information that arises during the course of the trial that you as a participant will want to know about, perhaps new risks, or you may have some particular questions that are pertinent to you and you would want to go back and have continued discussion with the investigator. So as I mentioned earlier, FDA has quite a lot of requirements for the informed consent process. I like to say that these are not just simply regulatory requirements, but they're requirements that were developed by the National Commission, which was a presidentially appointed, what I would call almost an ethics think tank, if you will, that was appointed after there were a series of egregious clinical trials that were exposed. And in the early 1970s, the National Commission came up with a set of recommendations for the federal agencies about what informed consent should look like, how it should ethically be accomplished. And we have actually incorporated those recommendations into FDA's regulations. And I'm going to present to you 3 of them today for our discussion, the 3 that we feel are the most challenging in relation to HIV cure research, but certainly our discussion can go beyond these if you feel that you want to raise other issues. And the first one is the description of any reasonably foreseeable risks or discomforts to the participant, a description of any benefits to the participant, either direct benefit or to others who may reasonably be expected to benefit from the research, and then a disclosure of appropriate alternative procedures or courses of treatment, if any, that might be advantageous to the participant. So in other words, if you're not in the clinical trial, what would you be getting as a form of treatment, or what would be happening with you? So I want to drill down in the next few slides. What I want to do is take each of those 3 elements that I just pointed out and look at them a little bit more in detail. And the first, the description of reasonably foreseeable risks or discomforts, FDA advises should describe the risks or discomforts of tests, interventions, and procedures required by the protocol, especially those that carry significant risk of morbidity or mortality. The possible risks or discomforts due to changes to a participant's medical care, that is, what's going to happen differently in the clinical trial than would have happened if you were receiving your care in the clinical setting. What are the risks of the drug itself? And what are the common and serious risks of the drug, and what might be done to lessen these risks or discomforts? And so, of course, we don't feel that the informed consent should in any way understate the likelihood, severity, or duration of these reasonably foreseeable risks and discomforts. And it may actually need to include information on foreseeable risks to others, such as to a fetus or to someone because of an infectious possibility of infection to others or radiation exposure to others. And then the description of benefits, we feel, should be clear, balanced, and not overly optimistic or overstated. We don't want to encourage people to be part of clinical trials by promoting the clinical trial, we want it to be a very balanced approach to the description of risk in the informed consent document. And again, it should describe the benefits not only to the participants in the research, but to others if there are any. And if there aren't any benefits that are direct, that should be stated clearly as well. And here I want to pause and describe what the therapeutic misconception is. This was a term that was proposed in the early 1980s, and it was proposed by Dr. Paul Applebaum and others. Dr. Applebaum is an eminent psychiatrist. And what he noticed is that people were confusing the intent of clinical research. And they were thinking that clinical research was really being designed to provide medical care. And in some circumstances, that certainly can be true, but there are very notable differences between clinical research and clinical care. And that is that clinical research is ultimately designed to answer a series of specific scientific questions or to meet a series of specific goals or objectives. And the care that a person will obtain in that clinical research protocol is dictated by the protocol itself. Whereas the medical care that a person receives as part of in the clinical setting delivered by his or her own physician is tailored to what would be the best medical care for a given patient. And so we tend to use the term therapeutic misconception when people believe that they're coming into a clinical trial to get the best medical care they can for themselves, rather than they may get the best medical care they could get as part of that clinical trial, but the overall objective of that activity is to increase scientific knowledge and the objectives of the trial. So finally, the last element of informed consent that I wanted to describe for you today in more detail is the description of alternatives. Participants should be made aware of what the alternatives are to their entering research, if any, that might be advantageous to them. It should include, if any, current medically recognized standard of care, approved therapies, or other forms of therapy, for example, surgical care or supportive care. And it should describe if participation in one clinical trial prohibits participation in another clinical trial. In other words, being enrolled dually in two clinical trials simultaneously. So today, in particular, we would really like to have your perspectives on and this is all within the context of HIV cure research. How should the informed consent clearly communicate to you the purpose of the research, the potential benefits, particularly if there are no direct health benefits for the participants, and the potential risks, particularly if there is very limited understanding about those potential risks? As Dr. Irony said, as the product moves down the trajectory of development, our understanding about its risks as well as its benefits may change. And so when you're in the very early phase where you've only introduced the product in animals, we don't know always what the risks are for human beings. So we want to understand how that can be best communicated to potential subjects, potential participants. And then finally, is there any other information that you think we would find helpful? I've included my contact information and the contact information of my office. We receive queries to this particular website if you have any. And I've also included a public access to our webpage, which I think you would find very interesting because it has lots of information that's helpful to clinical research participants as well as investigators and sponsors. Thank you.
Thank you, Dr. Goldkind. All right, we're about ready to get started on our discussion, but I'm going to do two things first. The first is there were some comments on the web this morning that pertain to the morning, and so I just want to make sure that those get summarized a bit. Well, while we're doing that, I'm also going to ask my colleagues at the table to reintroduce themselves because there are some new faces up here, and we also have some new faces out there, so it would be helpful if we could just go down the line. Please. Oh, you already introduced yourself, so maybe.
Damon Deming, I'm a virologist with the Division of Antiviral Products.
Jeff Murray, Deputy for Antiviral Products.
Kim Struble, Medical Team Leader, Antiviral Products.
Debbie Bernkrant, Division Director, Antiviral Products.
Celia Witten, Office Director of the Office of Cell Tissue and Gene Therapies at the Center for Biologics at FDA.
Ilan Irony, I'm the branch chief of the General Medicine branch in the Office of Cellular Tissue and Gene Therapies at CBER.
Adam Sherwatt, Medical Officer, Antiviral Products.
And Ed Cox, Director of the Office of Antimicrobial Products within CDER, FDA.
And while Andrea is summarizing these, if I can ask the panel members for this afternoon to come up, and we are getting one more chair from you because thank you. I guess I don't count very well. Looked like there were more chairs up there than there were. So.
Hi, so I'll just briefly summarize the webcast comments we received from this morning. We have a few folks who commented that about the side effects from HIV and taking HIV meds. So someone commented that taking HIV meds when dealing with nausea from chemotherapy is incredibly horrible. We have someone else who said that their dreams for HIV treatment would be no food restrictions and long-term dosing. And the most debilitating side effect for that patient would be intractable neuropathy. We also have someone who mentioned that the biggest issue is lipodystrophy in the form of facial wasting. And he provided some more details about that, about the treatments using facial fillers and that they're prohibitively expensive and out of reach for most. And that this issue is still huge in terms of stigma, self-esteem, depression, and isolation. So I think that's something new that we didn't touch on yet. And then just two other comments about developing long-acting injectable treatments and monthly visits that would be required. So this person was interested, maybe we can work this into the discussion, or if patients have comments on this, they could submit it to the docket. But this person was wondering how best they could support patients for monthly injections, and if patients would be willing to receive monthly injections versus daily pills and the impact on their lives. And then finally, we had a general comment about the context of this meeting and its relevance. And this commenter said that it's important to understand the importance of these issues in a global context and not just the U.S. And this person provided more details about the conclusions about today's meeting will have wide influence internationally, that this is a start, but it's important to also reach out to others.
Thank you, Andrea. Well, I'm not going to spend too much time going over the discussion format, but again, we have some new folks in the room, so I do want to make a few points about our discussion. Our discussion format will be very similar to this morning. We will first hear from a panel of patients and patient representatives. And again, when we say patients, we are talking about people living with HIV. And when we say representatives, we mean caretakers, loved ones, and advocates who speak on behalf of patients and who can provide comments on that fit the patient's perspective. Again, we're really focusing on getting the patient's perspective on the issues that we're discussing today. So we're first going to hear from the patient and patient representatives. I'll introduce them in a minute. And again, the purpose is just to set a good foundation for our discussion, to give us something that we can react to and discuss further. And then we will be moving out into the audience and opening it up like we did this morning, which is much more of a free discussion. So periodically, we're going to try our experiment again. Periodically, we will invite in-person and web participants to respond to some specific questions. Again, this is just a discussion aid. It just helps us understand the perspectives in the room and see how many participants share a particular perspective without needing a raise of hands or anything in the room. In person, we'll use the response clickers, and if you haven't gotten a response clicker and you are identified as a patient or patient representative, could you please raise your hand? Okay. All right, so Andrea will take care of that. And the web participants can respond through the poll through the webcast, and we got feedback that we went really quickly through those, so we'll try to give you some more time. And regarding the webcast, if you're just joining us on the webcast for this afternoon, we have about 180 to 200 people on the webcast. No, 100. We have more than 100 people on the webcast. Can't give an exact number. And you are very important to this meeting as well, and we are talking to you and we are listening to you. Even if we can't summarize all your comments in real time, we are taking them. Use the comment box and share your experiences as well. And for all of you, for those in the room, on the web, and others, we encourage you to provide your comments to the docket that is available on our meeting webpage. You can add on to what was talked about today. You can react to what you heard. We will be taking all of these comments and reviewing them, and they will ultimately get into our final product, which is a report on what we heard today. So a few ground rules. We really, again, want to hear from patients and patient representatives. There are a number of other stakeholders here from industry, other government agencies, etc. And as I said this morning, we hope that you find this as useful as my colleagues up here. But for today, you are in a listening mode and asking not to contribute to the discussion. There is an open public comment, and we will take until the next break. If you want to make a comment in the open public comment, please see the registration desk. Our discussion today will focus on the understanding the common ground on important issues. And we fully recognize that there are a number of important issues regarding HIV care and the support of patients, people who live with HIV. We will be trying to stay to the questions that we are trying to discuss today, the participation in cure research and the informed consent and how to clearly communicate about that. Again, there's a public comment if there's something else that you want to comment on. And if you haven't gotten a feedback form, we encourage you to grab an evaluation form and provide your feedback. It is completely voluntary, but we appreciate the feedback we get on how to make these meetings in the future as effective for everyone, for us, for the patient communities, and for other stakeholders. So thank you. Okay. So with that, we're going to test the clickers again. And we're going to ask the same demographic type questions that we asked this morning because we do have new folks here, and I guess practicing the clickers never hurts. So if we could have the clicker questions come up. While we do that, can we have the front panel members introduce themselves? I'll let you introduce yourselves.
Good afternoon, everybody. I'm Matt Sharp from San Francisco. I'm a long-term HIV treatment activist. I've worked with several AIDS service organizations, been on many advisory boards for pharmaceutical companies and also for research institutions. I'm currently doing independent consulting work, and I am no longer in San Francisco. I'm in Alameda. Had to say that.
Good afternoon. My name is Murray Penner. I'm the Deputy Executive Director of the National Alliance of State and Territorial AIDS Directors, or NASTAD. We are a membership organization that represents state health departments and their staff in HIV/AIDS programming as well as viral hepatitis programs. all the way from prevention through access to care and treatment, primarily through the AIDS drug assistance programs. I'm a person living with HIV and AIDS since 1986, and I've been in this field since 96, but it's only recently that I've become much more involved in sort of the advocacy community. Up until that point, I was doing a lot of on-the-ground work at a local health department.
My name is Jeff Taylor, and I've been living with HIV for over 30 years. I first got involved in the first AZT trials back in the 80s and have been involved in clinical research ever since. I was on the AIDS Clinical Trials Group, community constituency group, and currently I work with a group called the AIDS Treatment Activist Coalition, serve on the newly formed CARE Collaboratory Community Advisory Board, and I'm on the Antiretroviral Guidelines Panel. And I live in Palm Springs, California.
Hi, my name is David Evans. I'm a patient representative here from Project Inform in San Francisco. And where I come at this is that back in 1989, some eventful things happened. First, I came out, then I got a boyfriend, and he told me a week later that he was positive. Shortly thereafter, 2 of my closest friends died, and then my cousin came out as both gay and positive and had 30 T cells and pneumocystis pneumonia. And as a result of that, I joined ACT UP, which was a direct action group that still fortunately exists in some places. And shortly thereafter joined Project Inform's treatment hotline, where we answered thousands of calls from all over the country for people who are desperate for information about treatment. And the one thing I just want to say about that activist background and doing this so long is, in one way or another, we've been talking about a cure for a long time, but we didn't use the C word. We called it pathogenesis. We called it other things. And I just think it's important to note that we've been doing this for a long time, but it's great that we can actually get to the point where we can use the C word.
The C word. Okay. Hi, I'm Linda D., and I'm from AIDS Action Baltimore. Let me say all these little hats. I think that I probably work with most of the groups that everybody's talked about: AIDS Treatment Activist Coalition, the Community Constituency Group of the ACTG, the CARE Collaboratory. Worked with the FDA on a number of things, including their advisory panels, industry advisory boards, Hopkins Advisory Board for a very long time. Just invested. I've been doing this. My organization was formed in 1986. 87 was actually incorporated. And my husband died as a result of AIDS in 1987. And so many friends that probably as many people as in this audience. So I'm sick of this, and I want this to go away. And I know that the only way that that'll happen is if we find a cure for this. And I've worked with all of these people for many years, a lot of the people that are in the audience for many years. And it's very heartening that we're getting to even think about the cure word, let alone say it, and working in that regard. And I really would like to applaud the agency for getting to these questions and these points from not only a patient perspective, but in a timely way, so that we can really, have more of a chance to prepare for this than we did the first time around. So we're not knocking the doors down now. We're working on this together.
Okay, thank you very much. So let's ask those demographic questions that we asked this morning. The first one is an easy one. Do you live within the Washington, D.C. metropolitan area or outside of the Washington, D.C. metro area? If you got the clickers, it's 1 or 2. Okay. Can we have the... We are 50-50 this morning, so it's nice to see so many people travel from outside, and we really thank you, because this is quite an effort to do so. So our appreciation always for those living in town too, but our special thanks to those who have traveled here. Okay, let's go on to the next one. What is your age? Younger than 25, 25 to 34, 35 to 44, 45 to 54, 55 to 64, or 65 and better. Okay. Again, we have a distinguished crowd here today, so it's great. It's great to see all this wisdom in the room. Okay. Can we go on to the next one? Are you male, female, transgender, or prefer not to answer? Just like this morning, we have about the same split. Okay. Was it 50-50 this morning? Oh, okay. I'm so. Thank you for correcting me. You guys keep me honest. Okay. Have you been diagnosed as having HIV infection? Okay. So 2/3 of patient and patient representatives here today who've answered the question. Can I ask Andrea to, are the numbers available for the web?
Yes, about 70% answered no out of 20 responses.
Okay. Oh, I would have to do math for that. But we've got representation on the web as well. So we encourage those on the web to keep your comments coming in. Do we have one more? Yes, for persons living with HIV, how long ago was your diagnosis? Less than 2 years ago, 2 years ago to 10 years ago, 10 to 20 years ago, more than 20? I don't know or prefer not to answer. Okay. Again, a lot of wisdom in the room. Okay. And then I think there's one more demographic. Let's see. Yes. For persons living with HIV, how many different types of antiretroviral treatments, ART regimens, have you taken? And please include your current ART regimen if you're on one in your count. If you've never taken any ART regimens, you're currently on your first. If you've taken 2 to 3 different regimens, more than 3 different regimens, or if you're not sure. Okay. Lots of experience too with different treatments and different regimens. Okay. Good. Thank you. This really helps us get an understanding of who's in the room and what kind of perspectives we're getting today. So I thank you for answering those questions. We're going to have more questions throughout the afternoon, but enough of my talking. I'm going to ask the panel members to go down, and I might start with David. I understand that you have some data to share, so we'll start with David, and then we'll work and go throughout. Now agendas are what they are. We're a little bit behind on the agenda, so if you could stick to your comments to 2 to 3 minutes, and then we'll go out and you have plenty of time after that. So, David, thank you.
Sure. So one of the reasons that I wanted to be here today is that a really fantastic activist named Nelson Virgil and I conducted a survey of people living with HIV about a year ago about cure research. And one of the things that I want to say about the survey is that we did so based on the belief that people really would want to show up for these trials, even though they might not get any benefit out of them. And we fielded the survey between December of 2011 and early 2012. Within 6 weeks, we got 2,100 people to answer the survey, which for an online survey that we did for free and begged and borrowed and stole any kind of advertising that we could get, that's a pretty high response. The design of the survey was such that we made people have to answer every single question. So we got 100% completion on the survey response. I don't have time to describe all the demographics, I can say that despite really extensive outreach on our part to groups that are serving women, people of color, and people who are more recently diagnosed, our demographic looks a lot like the people who are here today, in that it tended to be a little bit more male, white, older, and having lived with HIV for a very long time. We also asked people before they answered the questions to read a primer that was, I would call it medium length. It wasn't short, but it wasn't really long. And it described a lot of the risks of the kind of cure research that we're engaged in right now or soon will be. We couldn't be sure that people actually read it, though, before they answered the question. So we can't say for certain how much they really understood the risks when they answered the questions that we did. But when we asked people to state how motivated they were to participate in a study that would benefit others but might carry risks for themselves, 88% reported being at least somewhat motivated, and 24% reported being very motivated to participate. Granted, there's a tremendous risk for what they call social desirability bias. Of course, everybody wants to help somebody else. And so that could be affecting the results a bit. But we still felt that was a pretty profound response. We also asked the question in a different way, and we received a very similar response. We asked people what their willingness to participate in a study would be if it definitely would not benefit them, but it might advance cure research in general. So a little bit more diffuse, not will it help people, but it'll advance the scientific field. And there we still had 81% reported being at least somewhat motivated, and 16% reported being very willing to participate. But I think that there are other benefits to participation in a study beyond altruism, and there are things that I think we have to really be careful and keep in mind. And we'll be talking about some of those things. But one little piece of data that we did when we did a deeper dive that I haven't talked about before, is that when we looked at people who were very motivated to participate to benefit others, they were also very, very motivated to benefit themselves in a study. And so I think that if we're depending on people's altruism, we also have to be very careful not to in any way play on any misperceptions they may have that they're going to benefit from a study when they actually won't. So we'll talk more about that.
Thank you, David. And then we'll start here with Matt and address the rest. And let me just, I didn't remind us because the panel will come back later after a break later this afternoon. So right now we're just focusing on the participation in cure research, what you believe are the benefits, what would motivate you to participate or not in an HIV cure research study, what risks you would find unacceptable, and why. And in particular, if you are asked to stop any HIV medications that you're currently taking, how that would affect your decision to participate. So Matt.
So I have some prepared statements if you don't mind. So I've been a participant in dozens of clinical trials over the years. I was actually first in every first-in-class trial of almost every antiretroviral drug class, except for nucleoside analogs. And I survived, obviously, but unfortunately developed resistance due to sequential monotherapy. I was always most interested in host-targeted approaches to HIV, but as you all know what ended up taking precedence. So I needed other options to buy more time, as my immune system was not up to snuff. And I am in that older HIV population at risk for inflammatory-related comorbidities that we talked about this morning. I participated in invasive trials, such as Dick Hong's thymus transplantation trial. I was in the first injectable Fusion Toro trials. I was in the first placebo-controlled human growth hormone trial. I was in the placebo arm at first of that trial. And most recently, I entered the Sangamo zinc finger nuclease gene therapy trial. So mostly I really had no choice to be in all of these studies. And despite my highly resistant virus, I've been undetectable for over 5 years now, and my T cells have doubled, remaining that way 2 years after this zinc finger trial. So I believe I'm really here as a result of being in clinical trials and an aggressive approach to fighting HIV from the very beginning. But the motivation in entering these cure-related trials is different today than what it was in the early days of the epidemic, as you all know. People needed treatment to buy time to live and survive. We have to do better today. There's much room for improvement in the care and treatment of people with HIV. This is something that we may have already been discovered through Sangamo's first gene therapy trials, where functional cure may not be likely, but additional immune recovery certainly has been shown in a few patients in the first cohort. So there's a lot of people out there like myself who want to take these risks and where there's no benefit because we want to live to see a cure. We want a cure for ourselves, or we want at least a functional cure. And then, as David said, many people will enroll for altruistic reasons. And at least I know in the gay community, it's known that people want to give back in the form of clinical research to help their own. When I go out and talk about a cure in the community, I always ask how many people would want to be involved in a cure-related trial, and almost every hand goes up in the room. But there are risks. And I'm a very different patient. I've been willing to risk a lot to be in these clinical trials. In the thymus transplantation study, I risk an invasive surgery and transplant rejection drugs. In the gene therapy trial, I obviously took a lot of risks with the manipulation of my own CD4 cells. Everyone is individual, though, and the risks they take might be more or less than others. Everyone needs to weigh their own informed decisions against the risk and benefit, obviously. And as the treatment guidelines state, everyone is individual. I feel that since there is going to be much variety in cure-related research, setting a standard of what is too risky, or on the other hand, not in-depth enough to get a clinical outcome won't really move the field forward. It's for this reason that the scientific and community collaboration is critical, so that each risk and benefit is weighed carefully as we go in a step-by-step manner, and everyone learns, and eventually we're all going to win. And I guess I'll stop there.
Thank you very much.
So, much like Matt, I've been infected since 1986, and in the very early days of the epidemic, as you all know, there were few treatment options, and so I was fortunate enough myself as well to participate in several clinical trials. And at the time, I was really looking at it from a very personal and a very selfish motivation, because I wanted access to what I knew was very promising in terms of options for therapy. Obviously, we are in a very different place now, and then I think that even at the time, I felt like there were probably a lot of risks that I didn't know the answers to, but I was willing to take those risks based on not having other options. We are at a very different place today, and so I think the whole motivation, and David's statistics were very interesting to me and actually not terribly surprising, because I think a lot of people do believe that there is really a good thing to be had by participating in these, but that therapeutic misconception that we heard about earlier is something that I think is in the minds of patients that are considering getting into these things now. One of the things that I would like to see if I were going into a clinical trial now was that there would be some kind of benefit for me, and I would get maybe more monitoring than I would, and more deeper dive into my, what's functioning inside my body than I would get if I just go to my HIV doc. So I think that it's a very different environment now in terms of what we look at with giving informed consent and having participants in cure research. For myself, I would really want to know the benefits of the scientific benefits and the risks that we believe are in place very clearly, without having to go through a 5-hour process with 9,000 different pages of something I didn't necessarily understand. From risks that I would find unacceptable, I think, I'm very used to the sort of the nausea, the fatigue, and the diarrhea. Of course, some of that could be that I'm getting older, so you never know which is caused by what. But nonetheless, some of the more immediate risks or symptoms that you might experience are ones that I would be concerned about to try to minimize, whereas some of the longer term risks I probably wouldn't be quite as concerned about, even though I know there could be a risk for cancer or other kinds of longer-term events, blood clots, seizures, etc. I think I would be less worried about that than I would be about the day-to-day kind of interactions that medications have on me. And then lastly, I think being asked to give up my treatment regimen that I've worked very hard to get something that works, number one, and two, doesn't interrupt my daily flow, is a real barrier in terms of participating in cure research. For myself, I'm not sure I would do it. Giving that up and having the risk of, further complications as a result of not staying on medications. I've taught myself over many, many years how to stay adherent, and to change that whole regimen up might be difficult for me to make that leap. I'll stop there.
Thank you, Murray.
Well, like Matt and Murray, I started early on, basically for the same reasons, to get access to drugs to stay alive. And, I think it's very different now, but back in those days, most of the trials were placebo-controlled. You got the drug, you didn't, and then they waited to see who lived or died, which is a kind of grim way of measuring efficacy. Unfortunately, we moved beyond that. But I think as we move into this arena, we're going to be back in those situations where there is no benefit in the control arm, or even in the treatment arm for that matter. And I'd like to share an anecdote about a couple of trials I was in where I didn't receive benefit and actually had some harm, and kind of how I got to that place and how it shaped my perceptions. One of the early ACTG PCP prophylaxis trials was comparing Bactrim, Mepron, and aerosolized metamidine to see which was most effective at preventing PCP. And I started on Bactrim, got the horrible rash. Same thing with Mepron, so was finally put on aerosolized metamidine. And as the study progressed, it was becoming more and more evident that that was not the optimal arm to be on. And my private physician came to me and said, you're on all these different antibiotics for all these things. I could desensitize you back to Bactrim. I've done it with other patients. I'm sure I could do it with you. You'd take fewer pills, and it would be better at preventing your PCP as well. And so, I was presented with a real dilemma. I went to the study nurses and said, my doctor has told me this. And they said, you're right. This is an option for you, and we're not going to tell you what to do. All we can tell you is that, at our site, nobody has broken through with aerosolized pentamidine. They've done fine. You've got 6 more months in the study, and the decision is yours. So being, the altruistic individual that I was, I said, what the heck? What's another 6 months after 2+ years of being in this trial? Well, sure enough, with 2 T cells, I developed a PCP. Both my lungs collapsed, and then I nearly died. But I pulled through. And, it was a valuable lesson learned. And would I do it again? Maybe not. I think I might have placed my own health as a priority because I still have lasting lung damage as a result of that. More recently, I was in a vaccine trial that had a structured treatment interruption. And I did the interruption. I was unfortunate enough to be during flu season, and I got the flu the same week I stopped my meds. So I think I really skewed their study results. And when I had to go back on, the regimen, I restarted my original regimen, and for some reason it didn't work. We couldn't find any evidence of resistance, fortunately, but they had to throw more pills on top to make me undetectable again. So I think, again, it's a personal example of where it didn't work, and I didn't get a benefit, and I would probably not be eligible to do a structured treatment interruption again. But even given that, in other circumstances, I think it's well worth it. I'm very committed to research. And, we're asked these questions, and there are no answers. It's just a lot of situations where it's highly individualized. Each person has to look at their own situation, sit down with their personal physician as well as the study doctors and staff, and figure out what's going to work for them, what their options are. If it doesn't work, examine all the scenarios one by one and decide what's best for them. That's all I'm going to say.
Thank you very much. And Linda?
So, I'm struck by something that on the care cab, the doc, the PI of the study came to us and gave us a letter from somebody from the North Carolina area who was sorry, who was a heterosexual who I think he may have said that he had all these religious issues, and I think felt very guilty that he was HIV positive and that he might have infected his wife and all of that sort of thing. So he was begging to be put on one of these clinical trials because of his situation. Now, here was a person that had no idea that these trials couldn't help him really, that he would really have no benefit. So I guess the point that I'm trying to make is that we really need to be sure that people know what they're getting into. I mean, not everybody has been through what the panelists have, through the years of the epidemic. So people hear cure, in the United States, we're like, little blurbs of cure. So, okay, well, where can I get some of that? So, I mean, we really have to be careful, and I think it's the community's role as much as the agency's role to make sure that people know what they're getting into. And we'll talk about that more in the informed consent stuff. And what would motivate you to participate? I'm being involved with the Hopkins CAB and the CARE Collaboratory. I've helped them get study participants for their leukapheresis cell trial. And, I was wondering, because everybody that I thought of, because you have to sit there for 3 hours, was on disability and could really use that $50 that they got, So those are the kind of people that I got in touch with, and a lot of them signed up. And I talked to a number of the other docs there, and they have found exactly what David's study has found, that there are a lot of people that are really interested in being in trials just for altruistic reasons. And most of those people are not on disability, at least in that little Hopkins group, So I guess people have different reasons for participating. And what would be an acceptable risk? I just finished a clinical trial in HCV, and you get these pages and pages of stuff that say, well, this could happen, that could happen. And I really often wonder, again, if people really understand what some of this stuff means. They either want to do it or they don't, whether they pay attention to what's even included in the informed consent. But with HIV cure trials, it's going to be so important that people understand that, this could really affect where you are now. So it's I think important to look at people in different groups. Now, a year ago, I would have said, well, maybe if I was newly diagnosed, I might be a better candidate for this. I might not be as big a chance for me to get off my treatment, or I wouldn't have a regimen like Murray described that has taken him years to get right. But now, then you have this new data that says, well, if you can get treated early enough and stay on just enough amount of time, that you might be, essentially able to go off treatment. So maybe that patient population is not the right patient population to start. If I'm trying to think about this as how I would think about it. So maybe the patients in the middle are the best ones. I don't know, but somehow all of this information has to be communicated to people to be sure that they understand, what the risk really is. Or you could get cancer. Well, what does that mean? How, when, how much? You see people that still smoke. Well, I won't get cancer. I'll even, how you've heard people say that, I'm sure. So if we could somehow do a better job of actually defining what risks we're talking about, and if it's a risk of a disease, if we can get a little bit more clear instead of this, and I'm sure we'll talk about this more when we get to informed consents, but a lot of those documents are really not to anymore, I think, not to protect patients, but they're to protect the IRBs and the institutions. So I think we need to get really, really drill down on what they should say, and I think we'll talk about that later, as I said. About stopping medications, that's really a tough one. It's really hard to have somebody that has really been sick for so long and say, okay, you're all right now, but the way to prove this drug is for you to stop taking your medications to see if or procedure that it's really worked. So, I think that it's going to be so important to be sure that people know what it is they're getting themselves into.
Thank you, Linda. Do any of my colleagues want to have a sort of a clarifying question, or Kim? Yeah.
I have a question, a follow-up question for Murray. I guess I was kind of struck by your comment because I thought the other way, that you thought that unacceptable risk, the long-term toxicity like having cancer, wasn't immediate for you. It was more the day-to-day types of toxicities and the known toxicities versus the theoretical toxicities. And I was wondering if others shared that same perspective, or can elaborate more on what they found was the immediate versus long-term, hypothetical versus known unacceptable risk for cure research.
Well, I just want to make sure that I'm clear. It's not that I wouldn't care about that. I think that's far less in my mind than would be what I have to deal with every day. I feel like we've all sort of experienced, you could die. I mean, I was told when I was diagnosed that I had 6 months to live, and here it is nearly 30 years later, fortunately. But I think it's sort of the down the road kinds of things I'm willing to take some risks on, more so than I am my quality of life now.
I can add to that a little bit just based on the survey that we did. First of all, it was interesting that people who had been positive the least long, who had the highest CD4 count, were the least likely to want to take part in these kinds of studies. The other thing we found that I thought was interesting, though, that runs a little counter to what Murray said, was that people felt very positive about their current antiretroviral therapy, and almost everybody was on it. 92% said they felt very positive or very positive, so very few people didn't like their therapy. And 75% reported only having mild or moderate side effects. And when we tried to analyze it and see whether how people felt about therapy affected their choice to participate in a clinical trial, what we found is that it didn't. Whether they felt great about their therapy or didn't like it, it didn't seem to matter. Granted, the people who didn't like it, there was a smaller number of people. What was I going to say? Yeah, that's it.
Anyone else from FDA? Okay, I'm sorry, Matt.
Yeah, just a quick another anecdote. In San Francisco, Quest Laboratories is performing some of the gene therapy trials that you may be aware of, and then they performed my cohort, and in the protocol they came back and asked us if we would like, after the initial infusion of the product, asked us if we wanted to stop therapy, and there was 8 participants in that cohort, and only 1 person said that they would. So that's just that example. And then there's a second study, a follow-up study that Sangamo is doing, that they are actually looking at a treatment interruption. And surprisingly, Dr. J is finding enough people to enter that and enroll in that study. So.
Celia.
Yeah, we had some comments about stopping a treatment, about the treatment interruption, and a comment, to stop to see if the treatment really worked. But in a lot of cases, the treatment interruption would really be more to stop to measure something to see how well it's worked, and not, not really with the expectation that it's worked. So I'm wondering if that would change any of your comments. And another question I have is about, risk, and the comments made, well, we'd like to know better what the risk is. But in at least for our products, what we have is just a level of uncertainty regarding what the risks really are, because truly the animal models are not good enough for to even delineate them, and the kinds of things that Dr. Irony talked about, like the risk of cancer with vectors that integrate, those are risks, but those kinds of things can come up without really being understood from the animal models. So I'm just wondering how the additional layer of uncertainty as it relates to risk would factor into your answer to number 3. So it's a question about number 4, your answer to number 4 and number 3.
Well, I think to piggyback on that discussion, I'm unlike Murray in that I'd be willing to tough out some short-term discomfort and inconvenience if I thought I was going to benefit science, but if I thought I might get cancer later, that might give me more pause. So I think people are very different, and you can't generalize in this, that it's going to be very different across the board, and you really can't make assumptions. But I think that if you ask enough people, you're going to find people to step forward for whatever reasons, as long as you ascertain that they're genuine and they really understand the risks.
Yeah, I think Linda mentioned earlier that it is very individualized, and I would certainly concur with that. And I think that your question about potentially stopping for a period of time to determine, the effectiveness and then have the particular options again, that might change my mind. I think some of the comments that I have made have been very broad and not specific to a situation. So I think that's certainly an important part of this, as well as the situation in which you're asked to do things. For me, I went through a period of time when I was determining regimens that worked for me, where I had a lot of diarrhea, where I had a lot of nausea, and I had a lot of vomiting, and those were terrible days. And that's why I think I'm motivated to say it the way I say it, that those things for me are really something that I don't want to repeat again. Whereas the longer term ones, yes, important, are less in my mind as those initial symptoms might be.
I may have said that badly about to see if it works, per se, is to see if what you're measuring is really happening the way you would like for it to be coming out. But, I guess what I was trying to get at is that maybe there are some populations for which that's less of a risk than others. For some of this stuff, we don't know what the risk is going to be. And if you asked 5 people the same question, you might get 5 different answers about that. So when I was looking at these questions, I went around in circles a number of times, and I thought, well, we're going to be a lot of help. We're going to give you more, you'll have more questions and answers by the time we get done. But I think if you think about it, really, if you think about the way we've developed drugs before, try and look at the populations where you can do the least harm. Try and make sure that people know what it is you're talking about. Try to define the risk as best you can. Be sure that you delineate that the person understands that there is no benefit here for you. That sort of thing. Are the concrete things the only concrete things that I could think of that were good take-home messages.
I'm going to interrupt this because I want to make sure that we allow the folks in the audience to also participate and contribute and build on what we're hearing. And before we do that, just to set the context, we have a couple questions for your clickers and for the folks on the web. This just lets us understand the experience with clinical research. So, have you ever participated in any type of clinical study related to HIV? Yes, no, or I'm not sure. Okay. So, the majority of you have participated in some form of clinical research related to HIV. Do we have the experiences on the web?
About 66% have not, 33% have.
Okay. And then another question is, have you ever participated in any type of clinical study specifically related to HIV cure research? Yes, no, or you're not sure? Okay. So more experience with the clinical research, just in general related to HIV, and less with cure research, which is not surprising, but it does help us understand where the perspectives are in the room. Did we get the numbers for the web?
Yep, the majority have not, about 93%.
Okay, great, thank you. So I know that my colleagues have more questions to ask, but what I want to ask first is if anyone here in the audience found anything said by the panel members really surprising. like it doesn't necessarily relate with your experience, or you would have thought something different, or in just surprising in general? Oh, and if you could state your name before you speak.
Hi, my name is Alex Garner, and I guess I was more surprised by what wasn't said, in the sense that as we had the demographic questions earlier before the discussion, I was struck by who wasn't represented and who isn't here, and who historically isn't represented in studies. And as we talk about the sort of vast experience that all of us in this room have, it's troubling to think of the future of the epidemic, which looks like, it's black and brown young gay men, and they're not here. So part of me wants to encourage all of us to sort of remember that, because it's critical to the work that we're doing, especially when communities of color have such a troubling history with federal agencies and studies and stuff like that. And when we talk about how people will feel inclined to participate in this study, how they're impacted by all these other elements of their life, class, race, sexuality, gender, etc., have an impact on that. And it's not just whether or not they're going to be feeling altruistic or not. It's all these other elements. So that's it.
Okay, thank you. Anyone else had any first reactions that were surprising? Okay, yes, Melanie.
Not surprising, based on the fact that a lot of these are long-term survivors in the beginning of the being infected in the beginning of the epidemic, and their desire to continue to live, to grab onto hope that it would transform from just staying alive to eventually being able to help somebody get a cure and not have to live long-term with having to take meds. I wasn't surprised about that. But for instance, in my particular case, with all my other conditions, I would not be willing to risk getting off the medication that I have been on, because I don't know if pulling the plug on that would cause something horrendous to happen to all my other conditions. So that's my perspective. Although altruistically, idealistically, I would be happy to be in a trial. But being a woman, there haven't been that many. However, I was able to be in one because I was on Depakote, and somewhere in Australia, Depakote was shown to deplete the reservoir of the virus, when you were suppressed. And so I was able to be in that one, but most of the other ones I was not.
So let's, go ahead. Was there another hand up? Let's just make sure that we have fully discussed the idea of the benefit. And we heard the altruism. Can I ask, David, did people have open-ended responses? The 80% who would participate, did they?
No, it was on a Likert scale, so it was very willing.
So does anyone want to follow up and build upon what we heard about any of the benefits or the perceived benefits of participating in a trial? Understanding that there's perception of benefits and then there's actual, real benefits. So if we could focus on what you believe are the benefits of the trial. If anyone wants to comment on that. In the panel, you can expand upon Matt.
So something I brought up this morning was, getting to the cure, there's going to be a lot of steps along the way that we discover. And one of those might be improvement in immunology an immunologic response. So that's one thing that could be a benefit.
Any question from FDA to follow up on that? Any other benefits? Yeah.
There are a couple of things that people might perceive as a benefit that we sometimes don't like to think about. And one of the things I can tell you is that when I was a broke student living on part-time money, I participated in research because it paid money. And another experience I've had as a clinical trial participant was going in, even though I knew I didn't want the therapy, but it would give me access to diagnostic procedures that weren't standard medical care that I had read would be beneficial to me. So just things that people think about.
You know, and a lot of the people that we have referred to Hopkins, a lot of them are African-American MSM, and a lot of them are on disability, and a lot of them were interested in the money that they got because they needed it. Now, I think we have to be really careful about the inducement there and whether we're, you know, getting people into things that are harmful or that they don't know what really the score is about. But I mean, I think that we really should. Yeah, well, let me just put it this way: when I talked to one of the doctors at Hopkins, I also asked about biopsies and does that dissuade people when he talks to them about what's actually required in a trial? And you know, he was saying, well, no, you know, that they're required to do anal biopsies in some of the prep trials, and people don't seem to mind, but that they really need to be compensated for what they're going through. And there's nothing wrong with that. I mean, I think that people are putting themselves on the line, and you know, some people need the money, some people don't, but it ought to at least be offered to people if we're sure they know what they're getting into and we're not using them in any sort of you know, illegal way. But you know, I think that's a real important thing in the beginning of this.
Anyone in the audience? Yes, go ahead.
Hi.
Yes,
Thank you.
and if you could state your name, please.
My name is Kevin Fisher. And one of the questions about benefit, one of the things where it sort of altruism overlaps with benefit is that there was a study that was very similar to the study that David referred to that was done in Holland with HVM, where they asked HIV positive individuals what they thought about a cure, why they might be interested in a cure. And interestingly, one of the things, as David said, a lot of people weren't necessarily concerned about their medications or toxicities necessarily, but one of the biggest concerns was about the future, like what will things happen in 20 years, and how will my life end, and and how will this disease affect me in ways that I don't quite understand. And one of the benefits of joining cure research was actually maybe to get a step towards that way. So this would be a case where you might join a trial even though it wouldn't necessarily benefit you, but you would actually, in some sense, push forward the general knowledge. This actually kind of goes to Matt's point. Push forward the general knowledge so that maybe one day people will have an answer to that, and they can maybe diminish some of that anxiety.
Are you talking about helping your future self or something
yes,
different?
Yes, a little bit. I mean, there's a general altruistic, but helping your future self in some sense. And so that's where altruism and personal benefit kind of overlap in a way.
Some people think too, and they did in the old days of the epidemic, that, well, I'm going to get my foot in the door because if I get in here, maybe I'll be considered for the next one and the next one, and I'll be in line for something that really will be beneficial 2 steps down the line. Now, a lot of times it disqualifies you from being in the next step, and people should be informed of that, you know, but I do think people think that way.
Yeah, I was going to reiterate that a little bit. That's sort of the motivation that I had early on, Linda, was really like, I'm going to get in. It's going to lead to the next place. It's going to lead to the next place. Of course, I was there weren't any options at the time, so it was like looking for that option that may be out there that ended up being there. But one of the things that I did find out, obviously, is my T cells increased, my viral load decreased as a result of being in this, and so I was seeing personal benefits. So the continuous feedback about consent about what's happening as a result of your participation in the study, at least for me, would keep me very motivated, thinking that there were other options that would help me improve my own life moving forward.
Do my panel members have any follow-up questions on the idea of motivation for participating or benefit? If not, let's move on to let's continue the discussion on the risks and the uncertainty about those risks. And we heard a lot mentioned up at the panel, and what I'd like to ask is, in the audience, if you had any thoughts that came to mind as you listened to the panelists about risks and you're thinking about those risks and how you might consider those for a cure research study. Nathaniel?
Good evening. I was listening and looking at the panel, and I thank y'all. I thank y'all for doing what y'all doing so I could be here. But I was listening to Murray, and I had almost forgot the days of diarrhea and throwing up and headaches. And I'm somewhat appreciative of your honesty. You know what I mean? Because what happens today, medicine scientists come together and they say, we're coming up with these clinical trials and we want this certain cohort of persons in the trial. Lydia, you spoke to it. A lot of them don't even read all that information that's in there, and they don't even look at the long-term effects that might happen down the road. I know I was one that didn't, and I got in the trial for what David spoke about, for the money at first. And then it dawned on me that I could get free health exams every 6 months. Not for my HIV, but for my high blood pressure, whatever else might be going on. And I was thinking, would I sacrifice the quality of my life today, get in a clinical trial for the future of somebody else? No, I wouldn't. And the reason I say no, I wouldn't, because the way the quality of my life is right now, no clinical trial helped me to maintain not a high level of my quality of life, but just a minimal level without me going back into the throwing up, the aches, the pain, the sleepless nights. And that's just the physical aspects. I ain't talking about mentally thinking I'm on an island by myself. So no, clinical trials have to give me more of a guarantee that I can still have some remnants of a mental and spiritual quality of life. Physically, I'll be able to deal with that if my mind and spirit is intact. So a lot of times, and I'm looking at y'all, and I want to say, when you have people in your life that support you, your life is more healthier and enriched. When I was single, my life wasn't enriched because I felt like every time I threw up, it was going to the worst, not fully understanding what was happening. But what clinical trials are not looking at, and Murray kept speaking to it, and it's so true. Once you get over a hurdle of physical discomfort and you realize that you are over that hurdle, you're not willing to subject yourself to that hurdle anymore for any particular reason without you getting a guarantee. You want a guarantee. And if anybody is involved and they have enough courage and integrity that's self-imposed, they'll say that. Don't care how much money is involved, Lydia. But your quality of life is more than money.
Thank you very much, Nathaniel. We have a follow-up question, or comment, I mean?
Not to that, but just to the risk versus the benefit.
Sure, yes.
My name is Fred Scheich. I'm with IFARA and ATAC. I am concerned. I really don't want to discount in any way David and Nelson's attempt to really get a good chunk of information from the community, and they did. The one thing I do care about is that when you take an arbitrary idea and you put it out there, it is that. But I think when you're talking about the reality of a trial, you have to really understand, that's what we're really going to be talking about next, I guess, the consideration of what is actually in that trial. What are the risks? What are the perceived risks? What are the discussed or potential risks that person could really evaluate fairly and honestly in themselves? And when you make that, and then the fact that you have a family or a work job that may be interfered with in this protocol, those are all those considerations that may make a difference. Altruism is a wonderful thing, and I tell you, most people have it, but it comes to a point where you have to take the reality in perspective. And that's the only thing I'd like to throw out there.
Thank you. Anyone else want to comment on risk? Yes.
Hi, this is Alex Garner again. As I'm thinking about the sort of risks and benefits associated with being in a trial, I'm reminded of how we worked so hard to change the risks associated with getting credit. Now you get very simple language, you understand what your risks are if you get a credit card, and we worked really hard to do that. I would suggest we do the same thing around clinical trials, so that people fully understand what the risks are in very clear language. We're also sort of assuming that they're at a particular level of health literacy, which isn't always the case. So I think those sorts of things help people understand what the risks are better, and they're more likely to trust the institution who's going to potentially put them at risks, and therefore more likely to want to participate and engage in that way.
Can I build on one thing that you just said, which is trust in the institution? And we haven't talked about that at all today. Would anyone like to comment on that point? Does that matter?
I'll take a stab at that as I've been thinking about after a couple of people have talked. One is that I think at least some of us up here have the benefit of a really informed treatment network and brilliant friends and really good doctors and a working relationship with the FDA and other agencies, et cetera, to the point where I think we probably do have a lot of trust, and I think that makes a big difference. Many of underserved populations, people living with HIV and AIDS, and it's been referenced here, don't have access to that, and I think it would be a lot more difficult to really have an honest dialogue with those individuals and have them involved in this. And we need those people involved in this type of research. And so that's one of the schisms I think that's out there that needs to be addressed, to develop some trust such that individuals do feel as though there is a good support system, as well as really clear, informed risks, benefits, et cetera, rather than just what's on a piece of paper that you sign.
Great. Anyone else want to build on that? Yes.
I'm going to basically say what Mary said in terms of like involvement in the process from the beginning, not just in the process after you have developed your informed consent until you have your conversation. I love all my comrades here from years, but here we don't represent the demographics of the epidemic, the experiences of all people with AIDS. So in the process, that will be a good way to start building that trust for the future. And more in Q research, that is going to be even more complicated.
I think when we come back after the break, I think we should spend quite a bit of time following up on how to engage the folks that aren't like those here in the room. So we can follow up on that. I want to ask my colleagues, though, before there's 2 more polling questions, so we're going to get to those before the break. But before we do any follow-up questions that you want to ask Sarah? Yes.
I have a follow-up question, but it might be something that we may have to revisit after the break. I've been hearing a lot of repeated comments. It's become a theme. There are 2 themes that I wanted to flesh out a little bit more. One is that it's an individual decision whether someone participates in research or not, and it's a very different decision from one person to the next, how risks and benefits are weighed. And so my question that I would have is, how do we help participants make that individual decision, and who is the onus on the investigator or the person who's doing the informed consent, or on the participant, or on their discussion together. So in other words, we could communicate information to the potential participant, and is it then that person's job to take that informed consent document to his or her healthcare provider or family members or community and figure out the risks and benefits, or should there be a second meeting after the original consent discussion with the investigator again for there to be additional discussion about the contents of the informed consent document? So I'd like to understand better what you think would be an ideal informed consent process in a sense. And the second thing that I've been hearing is that people don't read these very long, complicated informed consent documents. And so how can we communicate the information that's really salient in the informed consent documents to participants in a way that they will understand the information? So I think
I have a feeling we're going to be taking a break in a few minutes, and that's a huge discussion. So if we don't get to it now, we'll revisit that. But maybe one thing that I think could be asked is that's more directly related to the questions that will lead into after the break is that role of the personal physician, your own personal physician. I'm curious on how they shape your thinking and your motivation to participate in a trial. And I think that would feed into, we can revisit the question about informed consent then after the break. But anyone we heard a couple of folks up here, but anyone else like to comment on that? Melanie?
Yes, my name is Melanie Reese. I get my treatment at Johns Hopkins University, and they are a teaching research medical facility, and they push you getting into clinical trials. I don't know if they get a kickback, if they get so many of their patients in or not, but they don't really, they leave that informed consent to the investigator, and whether or not you get through the, what's that called? When you get screened, you go for the screening and then see if you could go to the next step, but they push it.
Anyone else have a different experience? Yes?
The regular physician of the patient.
Your regular physician.
Well, I mean, I have the same experience that everybody in terms of having access to good doctors. But now you made me think about the changing environment, with the expansion of Medicaid, the Affordable Care Act, Ryan White changing also in terms of how it provides the services, that when I think about the rest of the population that depend on that, and then probably they will go with primary care physicians that will have just 10 minutes to see these people, where will we have that conversation? How we support the physician to bring up that conversation? I just bring that up.
Great points. Okay. I want to make sure we get to a couple polling questions, and then we'll have a 15-minute break. Again, because that's what my agenda is telling me. So if we could have we've had a very rich conversation, and these are just two summary points, questions to get at the perspectives in the room. So can we have the next question? Okay. Now again, this is completely voluntary, and we're not going to hold you to these answers. But after hearing this discussion today, would you consider participating in an HIV cure research study if it was unlikely that you would gain any direct health benefit from participating? Yes, you would consider. No, you would not consider participating in a study, or you're not sure.
While people are answering that, can I say something about this question?
Sure.
You know, we've kind of lumped together trials here today, cure trials about whether you're going to get any benefit or whether it's going to really harm you in that you're going to have to stop your regimen or you may put yourself in danger. I mean, there are a lot of trials in the beginning that aren't going to really be like that. They all don't require that you stop your medication. So there's some where you have to sit there for a couple hours, let somebody take some blood, come back and that sort of thing. So you know, and it's going to be a very stepwise sort of thing. Now, there are other trials that do require more or will require more, but it's not all, you know, you're going to get hurt, you're not going to get hurt. It's not all that clear, I think.
You make a very good point. Perhaps for this would be the more intensive kind of trials. Okay? So there's a lot of folks would still consider participating in a study, although there are some folks who would not consider. Okay, Andrea, do we happen to have on the web?
50-50.
Okay. Good. So lots of good discussion. There's one other question that we want to put up. Similar question to the last one. Would you consider participating in an HIV cure research study if it meant that you had to temporarily stop taking your current HIV medications? Yes, you would consider participating in a study. No, you would not consider participating in a study, or you're not sure. And I understand that these two questions are really tied together because, you know, it's very related to the last question. Okay? Okay. Maybe it's not so related to the last question. This might be surprising to some of my colleagues. Thank you for your input. And on the web, for those of you on the web, we have over half who said no, you would not consider participating in a study, and about 20% of you in this, again, very small n, but are not sure about that. On the web?
40% yes, 40% I'm not sure, and 20% no.
Okay. Okay, there are a lot of unanswered questions then. Yes, go ahead.
Just a real quick comment on the risk thing before we go to break. I think it's really important that the FDA pay attention to exclusion criteria and what I call eggs in the basket. So with a lot of the earlier trials, you know, there were all these things like you can't have taken this class of drug, or you can't have done this, or you can't have done that. So a lot of what I think there's going to be a lot of concern on is, if I do the HDAC inhibitor, will I be excluded from the PD-1? And if I do disulfiram trial, will I be excluded from this? And that's in other words, it needs to be a really good reason if you're going to exclude under those types of things, if you know what I'm saying. And I understand that early, small, pilot, proof of concept studies need to be really clean. You don't want to muddy it with anything. But I think in the more advanced trials, you really have to justify very well any reason you're excluding people.
Okay. So why don't we take a break? We can revisit these